Stalin Raj, who is Associate Professor in IISER Trivandrum. His research area is coronavirus entry mechanism, receptor biology ACE2-DPP4, and viral pathogenesis and host interaction, emerging infectious disease. He is a well-known virologist and who has got several awards: Biosantex Research Mobility Fellowship, Franco-Indian Campus 2025, Crozier Foundation Award at University of Hong Kong 2018, and is postdoctoral fellow, University of Lille, Belgium, and also as a senior research fellow, Indo-Australian Project CSIRO earlier, and DST, he got several travel grants, and his publication has made a lot of impact in the virology field, and spanning publications from Nature to Lancet, infectious diseases and all, very high impact publications. With this very few words, I welcome my friend and collaborator also, Dr. V. Stalin, for presenting the talk.
Yeah, thank you, Dr. Rajendran. So it is a great pleasure to be here. And I would like to thank the organizing committee as well. So this is something, it's a self-signaling conference, and suddenly I got an invitation to deliver a talk. So my team at IISER Thiruvananthapuram is specifically working on entry pathway of coronaviruses, and also we are developing vaccines, indigenous vaccine platform for emerging pathogens. So since we don't have any, virology talk, I want to briefly give some introduction. Then I'm going to, connect somehow to my research on emerging pathogens. So, am I right? So, you all know about emerging pathogens. We no need much introduction about, emergence of novel viruses. Because of this fellow, so because of this COVID-19, we all know about coronaviruses at this moment. Not only coronaviruses, you also know about other viruses as well. So what is new here is we know that the coronavirus can cause severe diseases and cause pandemic as well, but that is not new. We already witnessed several outbreaks in the past as well. Even we all know very well about viruses, especially Spanish flu in 1918. Approximately 20 to 50 million people died because of these viruses. Fortunately, we could able to eradicate or control many of these viruses because of the vaccine strategy. You might not seen at least these polio cases. In the past, we have a lot of polio cases, or smallpox, or chickenpox, tetanus, mumps, and so on. We already eradicated, or we were able to control it. But what is happening in the recent years? There are a number of new viral outbreaks have been increasing year by year. Every year, approximately 3 to 4 new viral pathogens are rising and causing major outbreak. So one of the key things here, what you see here, the red is the known viruses often re-emerge, and the blue are happening new viruses. So one of the big question in the community is that what is the source of these viruses? You know that majority of these emerging viruses are originated from, is not a human, it is originated from the animal source. Animal source, you know that well-known example is that the HIV was originated from a chimpanzee, now become a human virus, and also cowpox, monkeypox as well, swine flu, influenza, and so on. So one of the key, if you look at some example, what is the source, how these viruses jump from animal to human, at approximately around 60 to 80% of the animal viruses are jump from, or cross-species barrier. So how do they do it? Basically, these are, we call it a spillover. These are the migrated birds. It's not belonging to us. During the winter season from Europe or America or China, the birds migrated from one continent to another continent, and they stay for a couple of months, then they return back. While moving here, what they have here, they also bring some of the pathogen. They can also spread between the species, and also they can they can cross the species to other species as well, and thereby they can also spread between interspecies, and also while go back, they can also take back to the other own country as well. So the other way around, if you treat pet animal as not a pet animal, there is a chance to reward as an infection. Maybe for example, if this kid is going to kiss, giving the treatment, it may not be a good idea. If the virus, if the swine already have any swine flu or hepatitis E, they have a chance of getting the infection to these cases as well. So in another way, if somebody else eat as a raw meat, I think if this is somebody is eating blood, having a contact with the blood, meat product or blood product is a direct contact, there's a chance of getting infection. As you see this in Africa, these are dead animal they are going to sell to, this in the live dead market. You see this fellow, what is he doing? He's eating a bat. You see this bat is a raw meat he eating. If there is a chance, if there's a virus in the species, easily can get transmitted to him. So one of the example what happened in Africa, you all aware about it, 2014 Ebola outbreak. One of the person ate bush meat. He got infected by the Ebola. So and then returned back. Then he died because of the Ebola. But no one aware about this the death is due to Ebola. But in Africa, there is a ceremony, death ceremony. If somebody die, so the whole family members or even the village members go and kiss the dead body. What happened this case, in the index case, basically, the first case he died, and everybody, or whole village people kissed the dead body, finally got infection from, to one person to another. Within a few months, the big outbreak, approximately 30,000 cases have been infected by this virus. And you all aware about it. It's another way of transmission. If another species, if we carry a virus, the vectors like a ticks or mosquito can carry the virus. They can also transmit it to, the vector-based disease. For example, if you look at that, this is a virus, you all aware about Zika virus outbreak. Zika virus, this is related, basically my work. What I want to tell you here, Zika viruses can cause microcephaly. Microcephaly means they cause a shrunken brain. If somebody has, got infected during the pregnancy, so then the child will born with, microcephaly. So what is interesting here is this is a two clades of virus. Both are Zika viruses. Some of the virus, there is we called as African and Asian clade. The Asian clade virus got some mutations. So the mutation, mutant causes neurological infections. That means this strain basically cause microcephaly. So what we show in this particular paper is that a lady returned back from Suriname. She got a Zika virus infection. It's a pregnant lady. So after 2 weeks of Zika virus infection, she got an early miscarriage. So then we took the villi and placenta and fetus. We showed that the virus found in the fetus as well. You see it in the villi and the fetus. We conclude that the early miscarriage is due to the Zika virus infection. So in another way of transmission can happen or jump this indirect way. You might know that we often see this kind of picture. Maybe guava eaten by bats. The bat's saliva might have some of the virus. Maybe if somebody else ate this guava, then we have a chance of getting infection. What you see here, this is the palm sap, by a bat, in Malaysia. So what usually do these bats? They drink, palm sap. While drinking it, they urinate in the pot itself. If somebody else next day drink like this, a chance of getting infection that, infection this route as well. That's the indirect way. So this we know that a couple of outbreak happened in Kerala, but we don't know what is the source. But these are all chances. So what I want to tell you, all this transmission is basically zoonotic transmission from animal to human. Transmission are quite complex. So whatever example that I showed you is not this chance of getting infection. You cannot think about, okay, if I ate, or if I close contact with another animal, I can get a chance. But this is a chance of getting. So what we know now is bats are considered as a reservoir host for many of the viruses. But bat to human contact is very limited. So what usually happens, the virus might jump from bat to another intermediate host. from the intermediate host, they can spread between the species, or either of the way they can get back to human. So what I'm going to talk about, the couple of examples on the 3 viruses, SARS-CoV-1, 2, and in the Middle East, respiratory syndrome coronaviruses. You all know about coronaviruses. I will explain. Coronavirus is also similar to other viruses, have a single standard positive sense RNA genome surrounded by a couple of protein we call as membrane protein, envelope protein. Another one important protein called spike protein. You all aware about this particular protein because you all vaccinated against this protein. This particular protein have a interest on it. The reason is this is the first protein is going to interact with the host cell receptors. So if you look at this particular protein, it's trimeric in nature. This consists of 2 domains, S1 domain and S2 domains. The S1 domain consists of 2 domains, subdomain we called as N-terminal and receptor binding domain. This particular receptor binding domain is going to interact with the receptors, but while this S2 domain helps them to fuse into the cells. So what I want to tell you here, the receptor binding domain of any of the coronaviruses almost same. So what you should do that is going to bind to the receptors. Even I tell you, whatever way they're going to bind it, they get mutate and evolve and phosphate a different variant, but still it's going to bind it to the receptor of its own. Okay, what we know now, the different coronaviruses uses different viruses. For example, human coronavirus 229E, we use aminopeptidase N of, and MERS coronaviruses use DPP4. I was discovered this one. And another one is SARS-CoV-1, SARS-CoV-2, and human coronavirus NL63. These viruses use ACE2. So what I want to tell you here, the human to animal to human transmission, the virus might use, the animal APN or DPP4 ACE2. That virus could be a prototype. That virus might make this spike adapted to the human DPP4 or human APN or human ACE2 to jump to the one species to another species. Okay, so once it binds to the receptor of a respective virus, binds to the virus enter 2 distinct pathways to enter in the cells. One is receptor-mediated endocytosis, endocytic pathway. Another one is direct membrane fusion. So upon binding of the virus, there will be a membrane-bind proteases directly cleave the membrane, then release the genetic material in the cytosol, or they enter into the endocytic pathway, either clathrin or caluolin-mediated endocytosis. Once it's there, they release the genetic material, they make a new copies and go back to the cells. What I want to tell you about in coronaviruses not new. But we know in 2019, but the first human coronavirus was discovered in 1937. We call it actually this is an infectious bronchitis virus, basically from a chicken. The first human coronavirus was discovered in 1967. It's a human coronavirus 229E. But we all came to know in 2002 SARS-CoV-1 outbreak. I don't know many of you aware of not. This was the picture was from the newspaper. There was a big outbreak was ongoing in China. Many people are dying because of this infection. What they found it that this was a new coronavirus. What we know, these are the coronaviruses, is already known coronaviruses, but this the one they found in 2002 was entirely different from the known coronavirus. People are asking questions. We never seen any highly pathogenic coronavirus in human being. How suddenly this coronavirus appear in a human population? There's a big question mark because we already know this coronavirus research group. Majority of the coronaviruses are actually zoonotic viruses. Look at this. One of the questions came in the scientists. So this might be a zoonotic viruses. This is a Chinese animal market. These are not for slaughtering, not for a pet. This is for slaughtering purposes. Okay, so this is this is a bat belly. They make a soup, and they drink it during some of the occasions, and they can find they keep animals in a market like this. So there's a chance of intertransmission, and also they can maybe the virus might come from this one of these species. So what they found, they found the animals, one of the animal called civet cat. The virus basically from civet cat and it jumped to human and caused a big outbreak. So later they came to know that real story. Yeah, actually chef was cutting a civet cat in the restaurant and he got infected this, from the civet cat. Then whoever comes to the restaurant give us a gift as a, as a disease, then they spread the virus between to human. At least 10% of the people being died because of this outbreak. So what we know after 10 years later, so originally the virus was from the bats. They used bat ACE2 to replicate in bat cells. This virus adapted to civet cat ACE2 by mutating the spike. So then from this civet cat, they jumped into human. So what happened, this human adapter strain, they can replicate in a civet ACE2, not on bat ACE2. So 10 years later, exactly 10 years later, so another coronavirus was discovered from the Middle East. Maybe this is majority of the work is from my side. So this particular virus I was discussing, SARS-CoV, now we call it SARS-CoV-1. The one we discovered is called human coronavirus EMC. Now this virus named as Middle East Respiratory Syndrome coronavirus. What we see that this virus is basically entirely different from this pathogenic SARS-CoV-1. It's from a separate clade and also cause human diseases more close to a bad coronavirus. So we were asking a question where this virus is going to bind it. Indeed, we found out this virus binding a new protein we called as DPP4, dipeptidyl peptidase 4, as a receptor for this virus using a lot of experiment. We proved that this is a receptor. And what we proved that this virus, bat also using the same receptor, DPP4, to enter in the host cell. So you may ask a question, why do you want to know about the receptors? Receptor is very important to answer many questions. One is you can develop animal models, diagnostics, therapeutics, and also you can study the pathogenesis and also transmission. So here I would say this is the lung epithelial cells, even tissue of lung tissue. You see this is cilia, this is non-ciliated cells expressing DPP4. What does it mean? If you look at the human lung respiratory system, you have upper respiratory and lower respiratory tract. Upper respiratory tract, you will have a huge number of ciliated cells. Lower respiratory tract, you have a non-ciliated cells. If the virus, if you express non-ciliated cells, that means the virus, you need more number of virus to get into the gut infection. But if the virus want to transmit like a SARS-CoV-1, we need more number of virus to secrete out. Am I right? But in the case of SARS-CoV-1 or 2, they replicate in the upper respiratory tract. They use in the ACE2. They can spread very rapidly between, throw in or excrete out, and also they can also take an in as well. So later we were tracing back where this virus is, circulating this species. Indeed, we found out that the camel is the reservoir host for this virus. So what we conclude now with a lot of studies, we show that the virus use bat as a DPP4 in best-to-go cells, and also they adapted to do the camel DPP4. From the camel, they jumped to human, and also the human adapted strain still able to bind to the human DPP4. So now, approximately 17 years later, we have a virus. Now, you all know about SARS-CoV-2. You see the SARS-CoV-1 is here, the virus that we discovered here. There's another virus. This is using ACE2 entry receptor. But what interesting is that there are several new viruses being discovered from bat or another species. Majority of them are using ACE2 as a receptor for this virus. Potentially, we can cause a future disease or outbreak. We don't know yet. There's a possibility. So I'm skipping to some of the things. I'm going to move into this. What we are doing we are asking a very simple questions like a cell signaling. Upon binding of a receptors, how do this virus enter into the host cell? What trigger to take internalized into the host cell? We all know about the receptor, am I right? One ectodomain, one transmembrane domain, and cytosolic domain. Upon binding of a ligand, the cytoplasmic tail can activate and trigger the adapters to endocytis pathway. So our question is, ACE2 have a cytosolic tail, is around 40 amino acid, which indeed, whether it is triggered to the endocytic pathway or not, we found some of the critical motifs, which is, endocytic fortification from the cytoplasmic tail of the ACE2. We were assuming that this indeed helping to internalize in the host cell. We also found this motif in different species of, the ACE2 cytosolic tail. And we made a different mutant. I'm going to talk about one mutant. We called a cytoplasmic tail deleted mutants. We generated mutant. We confirmed that the mutant is indeed expressed on the membrane of the cells, even if you delete it, like a wild type. And also, if you use the protein of recombinant protein of the spike, the S1 domain of spike, indeed bind on the membrane as like a wild type. So that means very clear. And also we could show that indeed they are also localized on the membrane. As you see here, this is a ACE2, this is a spike. The spike is on the bottom, ACE2 is on the bottom, the spike is on the top. Indeed, they co-localizes in both cases, either wild type or cytoplasmic deleted. That is very clear it is. Then we also, if you allow the spike to internalize into the host cell, indeed, with or without the ACE2 cytoplasmic tail, the protein is internalized and co-localized with the receptors. So this, subsequently, if you do a virus infection in two independent cells, if you transiently express this cytoplasmic tail, it is a receptor. Indeed, the virus goes inside the cells as like a wild type, and we clearly shows that the cytoplasmic tail is not essential for the entry of the SARS-CoV-2 or SARS-CoV-1. So what is even further, we also show for other viruses as well. Similar case for Middle East respiratory syndrome coronavirus, the DPP4 cytoplasmic tail is actually not, signaling was not essential for the entry of this MERS as well. Subsequently, if you disturb the dimerization of this, usually the DPP4 or ACE2 form a dimer. If you disturb them, the dimerization by mutating them, still it is make a monomeric form, what you can see it. Even if it is a monomeric form, still they bind and able to infect, internalize into the host cells. So meaning that either cytoplasmic tail or dimerization of receptor dimerization is not required. So meaning that the virus is a ACE2 receptor might be a primary receptor, may use a co-receptor like other viruses like a HIV and hepatitis C virus. These are the two viruses, well-studied viruses. They bind to the primary receptor. They use several co-receptors to enter in the host cells. I think these viruses also might use similar mechanisms. We already know that several cofactors play a key role for the entry of. We don't know which one play a key role. We are working on it. So moving to the next part, when you as is shown in the previously, with or without cytoplasmic tail, the virus internalized into the host cell. Am I right to go in? So this is a typical cell biology. If anything bind on receptor, they enter through the endocytic pathway like either clathrin and caveolin. From the caveolin, they can reach the early endosome. From the early endosome, there are, it's a kind of, sorting system. The cargo can sort and then send to either plasma membrane. There are two independent pathways. One is a fast recycling mechanism or the slow recycling mechanism. On the other hand, they can also go into trans Golgi or a late endosome or other organelles as well. So we want to know whether these particles, once you bind or the virus, enter through which route and how this trafficking is happening within a cell. We did the assays using different markers, for example, organelle-specific markers tagged with either GFP or RFP, and also we developed several different types of stable cell lines and to do the study. What I want to tell you that when the experiment is very simple, we express the organelle-specific markers along with the receptor, then allow the spike to bind it, then subsequently we allow them to internalize in the cells. So, that means the binding we kept it 4 degrees, then moved them 37 to internalize it. So then we read out where to go. So what we see is the virus, the spike protein along with the receptor internalize, enter and reach the, early endosome. You all know about this is nothing new, but it's a known fact is anything goes in, reach of the early endosome is an endosomal marker. and to form a multivesicular endosome, what we see here as well. This was found in all 3 viruses, even SARS-CoV-1, ACE2 also we could see it, and also we could see a co-localization, spike, ACE2 cause. So that is indeed expected, which is nothing new here. What I want to tell you, SARS-CoV-1 or SARS-CoV-2 or MERS-CoV, we could see the multivesicular endosome. The big question, maybe we use a different marker, I will skip it. So the big question is, from here, where do they go? Either they go to the Golgi or late endosome or recycling endosome or any other organelles. So we did an experiment. First we checked it whether the spike protein, whether to go into the late endosome, because there is a possibility they can go to the late endosome. So what we found is that very limited amount of protein is moving to the late endosome. It's not to the, as is shown in the previous picture, you know, there are more amount of protein lies in the early endosome, but very limited number we found in the late endosome. This phenomenon for both cases, SARS-CoV-1, MERS-CoV, and also SARS-CoV-2 as well. So what is found is very interesting is that we found a significant amount of protein localization in the late endosome, I think recycling endosome. So indicating that the protein might move to the recycling back to the late endosome, but this is also not reflect the amount of protein that we observed in the early endosome. So this is a similar thing we found it for the, even the SARS-CoV-1 or SARS-CoV-2 or even MERS-CoV also we found the similar phenomena. And also, but we didn't see any protein localization in the mitochondria, or I didn't find any endoplasmic reticulum. That is very clear. This protein, we no co-localization at all in the case of. So what we was found very interesting is majority of the protein was moved from the early to trans-Golgi network. So this was quite surprising because the proteins, majority of them, you see that in the case of SARS-1, this is a similar case. Even if you cytoplasmic tail deleted DPP4 or ACE2, they follow the similar early, and also we found the RAB and Golgi. We see the recycling endosome co-localization clearly shows that they go from the early, from the early endosome, they reach the trans-Golgi network. From there, go back to the plasma membrane. What is interesting is not only a spike of or viral protein, anything, if you put as a viral antibody, we use the receptor-specific antibody. The receptor-specific antibody also follow the same route. This is what clearly shows that this formed in the case of both, DPP4 or a specific antibody also bind and go to the trans-Golgi and go back to the plasma membrane. So what we could later we could able to high resolution and super resolution microscope, we can also pinpoint in each and every molecule, a single molecule level, we could able to trace back how this molecule, you see this is all spike protein is attached to the plasma membrane, and of course I'm not, due to the time, I'm not putting all the slides, but basically we could able to show how yes, done, so I'm done. So this is the way going. What I want to conclude is, okay, so basically the spike is going to bind on the membrane, then reach the early endosome, form a multivesicular endosome. From there, a small portion reach the RAB7, that means late endosome. Majority of the protein is reaching in the trans-Golgi network. From this network, either the protein might trans back to early, then go back to the slow recycling mechanism to reach back the plasma membrane, or they can go back late and go back to the early, or directly go back to the early endosome, late recycling endosome, they go back. So what is clearly shows that maybe what is interesting here is, we also did some drug assays, I'm not doing. So what is interesting for as a researcher, so you all know about SARS-CoV can cause respiratory infection, am I right? It cause lung infection. You see, this is the ACE2 expression different organs. You see kidney tubes and cardio, all kind of organs with high level of expression. If you look at this lung, very little amount of the ACE2 expression compared to all other organs. But we have organ failure, is very limited organ failure, at majority is actually lung infection. What does it mean? Meaning that the virus enter into the early endosome, they go to the trans Golgi, along with the receptor, they go back to the plasma membrane by escaping the degradation pathway. If we go this way, the lysosomal pathway, what will happen? The virus can be released, genetic material, but in this case, it's not going to happen, they recycle back. So why I want to tell you, if the virus use lung, bind the lung, there is a protein called TMPRSS2, is a serine protease, directly cleave the plasma membrane, they cleave the membrane, they directly release the genetic material in the plasma membrane, okay? But if the virus use the endocytic pathway, then they escape from the degradation, so thereby they simply recycle back. There is the reason we might not see much organelle failure during COVID, but very limited maybe. If you have high infection, there is a chance, but otherwise not. This is what we want to conclude it from here. So with this, I would like to thank a lot of people. I put some here, and this is my team, and also this is a facility recently we inaugurated, and also funding support from many Indo-EU grant and ANRF, DBT, Intramural Grant, ICMR, and DBT, IRFA, and so on. PhD fellowship from INSPIRE, CSIR, and also PMRF fellowship, and also other members from India and abroad, but Hackman from Erasmus Medical Center, Brian John Bose from Utrecht University, and Amit Avasti from, this is some of the vaccine studies and all we are doing it. So to this, I would like to thank everybody for the kind attention. Thank you very much.
Excellent talk, excellent research. We take some questions. 1, 2, first, second.
Yeah, very fascinating talk, no doubt about it. Also good pictures of your transfection and then coagulation patterns to be clearly visible. We know that ACE2 is one of the known receptors. There would be any redundancy as well with ACE2 or RAF5, which basically recycles the viral particles. So if suppose we have certain thing which can probably block RAF5, can we contain the virus replication endogenously?
RAB5?
Yep.
RAB5 inhibitors. So the RAB5, of course, we can't inhibit anyway, because the RAB5 is a main machinery, like a post office, but they collect all the letters and they dump into the local post office. From there, they're going to sort it, depends on the address. Same thing, anything come from out, they bring to the endosome, everything sort from there, then they decide where to go. So if you block it, ultimately all the endocytic pathway is going to block it.
Okay. Thank you.
ACE2 blockers. ACE2 inhibitors doesn't inhibit the entry of the pathway. That's what we also want to tell. This is just a ACE2 as just in a binding partner. So once it binds, other cofactors, we already know one of the cofactors, we are working on it. So you're going to bind it, and they take in, with the help of the one or two cofactors, they can take in.
Yeah.
So if they use the endocytic pathway, that's the reason I told you. Other organs have a ACE2, they bind it, but they can recycle back to the ACE2 recycling mechanism. They escape from the the regular route. So if there is a TMPRSS expressing cells, then no need of using this pathway. They directly cleave it and they release the genetic material within a cell.
So this was very informative for me as a non-virologist. So my question relates to your spike protein, which you said is a trimer.
Trimer.
Right. Can a monomer bind to the receptor? Only a trimer is required, because this will have a consequence on the epitope that you use for immunology.
Yeah, okay. We never use a monomer. We use a dimer. Dimer bind it. We never test monomer.
So then making an antibody against a monomer may not work against a dimer.
No, it will work.
It will work?
It's
Okay.
a monomer, antibodies work. People are, okay, now I will come back to the, sorry. People are expressing the protein in the prokaryotic system as a monomer. still they bind it. Yeah, true.
Stalin,
Yes.
fantastic talk, man. The question is this: the possibility of the virus binding on lung epithelial cells is minimal, looking at the number of binding sites possible. Other tissues have more amount of
Yeah.
viral binding possibility, right? But still the lung is what is affected. But why?
So that's what I said. Lungs cells, epithelials have TMPRSS2, is a membrane-bound, serine protease.
The last slide that you showed about the localization, what is it?
Yeah, yeah,
ACE2 or a TM+2 or both?
This this is ACE2 expression, not
ACE2.
TMPRSS.
Okay, all right.
We already know that many of these cells doesn't express TMPRSS.
So you
that
need-
means this, even if you bind, they can use the endocytic pathway.
Okay.
So this case, we know that they express a TMPRSS. This is well known.
But TM+2 is very much there in the testis as well, right?
Yeah.
So the virus entry through testis is not possible ultimately, right? Like you said.
Can you?
Like what I'm saying is that TM+2, you say is there in lung epithelial cells.
Yes.
That's what you said,
lung,
right?
Yeah.
But TM+2 is there in the testis also.
Yes.
Yeah?
Yes. But TMPRSS is not there.
In the testis?
Which one? Which one?
TM+2.
Yeah, then you will be get infected. If there is a TMPRSS2 plus ACE2, the infection is possible. Infection is high level. So I mean, what I want to tell you, if even the virus use the endocytic pathway, you need more number of virus to establish the viral infection. Okay. So in the case of less expressing, ACE2 even expressing the low level in the lung, just a binding, TMPRSS help them to, cleave or cut this, the spike and directly, help them to fuse into the plasma membrane.
Yeah, what I was thinking was testis is, I don't know about the other tissues. Testis I know has both ACE2
And
and TM+2.
Yes.
Both are there.
In that case, there is a, definitely will get infected. Yeah.
Yeah, that's what I was.
Yes.
Hello, sir. Nice talk, sir. A lot of good data. And also there is a protein called SND1. This protein is a component of RISC complex, RNA-induced silencing complex. It is an enzyme, ribonuclease enzyme.
Okay.
Simultaneously, it acts as a coactivator for SARS-CoV-2 replication.
Okay.
Do you think we can target that enzyme to prevent SARS-CoV-2 replication?
Replication, if you inhibitors, we can stop them, right? But see, so far, not only SARS-CoV-2, any viruses, other than HIV or hepatitis C, some viruses, so none of the drug molecules not helping, not very efficiently.
Okay.
So that's a very different reason, because they direct cell-to-cell fusion. What usually happens once the particle is there, then directly fuse to the another cells, they can start replicate there as well.
Okay, thank you.
I just have one small question. You said that virus take an endolytic pathway or recycling pathway, depends on the severity of the infection, right? So is there any other mechanism that it choose, or only severity of the infection?
No, this is what I said because of our hypothesis, okay? So what we said here, even not only spike, if you tag the antibody against the ACE2 or DPP4, is go back to the trans Golgi, then returning back to the plasma membrane. So because of that reason, we hypothesize that this is, could be why we don't get much, severe infection.
This is not known, right?
Not much known about it, but we are doing a lot of other works, even live virus studies. A lot of other issues are there to prove this, but we are working.
Is there any comparison with some other virus which is already known this kind of pathways?
Nobody, as per my knowledge, not related, this recycling not be. But we know about receptor recycling mechanism for other receptors. That mechanism we know. So because of that reason also we are speculating this is moving this route and go back. But that depends on the receptor. So, some of them directly go back, fast recycling, they go back to the plasma membrane. Some go back slow through the trans Golgi and go back.
Any more questions? So as we are ending this session, and thank you very much again for the illuminating talk, and our co-chair is.
Yeah. Hi, Dr. Stalin. It was a very informative talk. So I have one question where regarding the non-receptor mediated uptake. Is there any report of SARS-CoV-2 like ADE, non-neutralizing antibody?
So
dependent,
AD
is reported in this,
So this is actually AD is actually not related to other cells. The virus doesn't replicate in macrophages, am I right? So there are some people are talking about it, but I'm personally, I don't, agree with that. So the virus can go in the macrophages through ADCC, but I wonder whether it is able to replicate or not.
So in the late endosome, is there how it is actually evading the cathaspins and just going out like dengue? There is a fusion loop and fuses with the membrane.
Exactly.
So how it is actually escaping from that?
But this is typically is follow the route of ACE2 recycling. It's not related to virus or anything pathogen. So receptor has to maintain the homeostasis on the membrane. So whichever bind they taken in to go back to the plasma membrane through the Golgi network. So because of that reason they evade, but we don't know what any other say. Of course, these all study all markers and we
Yes,
stuff.
yes.
Am I right?
Thank you, Dr. Stalin. Thank you.
Thank you, sir, for the enlightening talk. Now may I now request Dr. Sundar Bhattacharya to present the memento as a token of our sincere appreciation.
Thank you. Jarajendran. Thank you.
By this, we have come to the end of the technical session 5. We sincerely thank our respected chairperson,